Hello student, I am Dr. Gaurav Kumar and today lecture topic is nanotechnology in the drug discovery process. So in the previous lecture, we learned about the bioinformatics and drug discovery process, how bioinformatics tool and computer-aided drug design can help to discover, to identify a drug molecule, to identify a target molecule, and how it can help in the drug discovery process. Also, we have learned about pharmacogenomics applications in the drug discovery and development. We have analyzed previously how an molecular diagnostic to find the SNP and as well as personalized medicine help to drug discovery and development process. So today's lecture objective is to understand about the nanobiotechnology, to analyze the significance of nanobiotechnology in the drug design, to evaluate the significance of nanobiotechnology in the drug development process. We will also examine the role of gold nanoparticle in the drug design and development process, the quantum dot for the drug discovery process. There is conjugate of the nanoparticle also exists, and with the help of the DNA and proteins, we can also help to the drug design and development process, how a nano chip is useful and beneficial for the drug design and development process. We will also analyze the microfluidic device that is also called lab on a chip, organ on a chip, liver on a chip, how this microfluidic device can help for the drug design and development process. So we will learn about the nanoparticle. So nanoparticle uses and the risk is associated with the nanoparticles we will investigate in this lecture. So nanoparticle is a very teeny particle and it has a diameter of about 1 nanometer to 100 nanometer. So it is a really very teeny particle exists, and we can also synthesize the nanoparticle, and we cannot, this nanoparticle cannot be visualized using a light microscope, simple light microscope, and the study of the nanoparticle is called nanoscience. And there are several type of nanoparticle which is have the useful property, and their key feature is the large surface area to the volume ratio. So as the volume decrease, their surface area is increased, which is the basic principle of the nanoparticle. So there is the several use of the nanoparticle. It depends on their surface area. So the nanoparticle can be used as a catalyst. So the catalyst, the exposed surface area, is the most important point of the nanoparticle. It needs much less of a material made of the nanoparticle than it has a bigger particle. So in the nanomedicine, nanoparticle is used for the medicine purpose also, and we will also analyze their roles. So in the nanoparticle like fluorine, fluorine is a type of nanoparticle, is so teeny, and it is used to deliver the drug around the body. It also delivers drug to the inside of the cells. The nanoparticle is also used for the development of electrical circuit. Some of them can conduct the electricity. So it is also used to develop the teeny computer chips. The cellular nanoparticle has antibacterial property. So when silver nanoparticle is used for the development of the surgical mask, skin ointment for the wound healing property also. So there are several applications of the nanoparticles in the drug discovery as well as drug delivery process. So it is a relatively new branch and its effect on our body is not fully understood. So there is some issues with the associated with the nanoparticle. So actually it cannot be sure that how it respond a physiological effect in the our body. So some people think that we need to regulate them more strictly and level anything that might contain the nanoparticle. So this is the precaution measure for the use of the nanoparticle. So nanotechnology is a novel branch and an emerging area in the and has a wide application in drug discovery process. So the application of nanobiotechnology has an impact on diagnosis and drug delivery system. So currently, researchers are starting to use nanotechnology in the field of drug discovery and development. For example, nanocrystal is a type of nanoparticle and there are other nanoparticle also, that is gold nanocolloid, magnetic nanoparticle, barcodes, nanobodies, dendrimers, fluorines, and nanocells. There is also some is not listed here, but carbon nanotubes is also belong to the nanoparticles. So these nanoparticles have unique advantage for the development of the drug and its discovery. That is, quantum dot and magnetic nanoparticle are being used for the barcoding of specific analytes, which are key component of the biobarcode assay. It can be a future alternative to the protein chain reaction also. So the term nanomolecular diagnostic is the use of nanobiotechnology at the molecular level to diagnose and the treat of the chronic diseases. So extending the limits of the molecular diagnostic to the nanoscale. The nanomolecular diagnostic field can meet the rigorous demand of the clinical laboratory and can be cost effective. So here is this picture illustrate about the benefits of the nanotechnology through the drug discovery process. So nanotechnology have a broad area and a wide area of application like improved understanding of chemicals at the cellular and molecular level. By using the nanotechnology, we can increase the throughput technology and we can improve the identification and validation of the target protein as well as drug levels. So the nanotechnology is also used to reduce the time for the identification of the new drug. It is also used for the reduction of the precious reagent required to screen the potential drugs. So the nanotechnology is also applied for the improvement of the visualization of the drug interaction. So this figure is only a symbolic representation. It does not consist all the related field and all the benefits of the nanotechnology. So nanotechnology has a wide and various applications in the drug discovery process. So we will see some application of the nanotechnology in the drug discovery process in the following lectures. So the nanoparticle have emerged as a promising nano platform for the efficient diagnostic and therapeutics, processing characteristics property at the nanometric scale. So the feasible immobilization of the specific ligand on the surface of the biological site have become ideal candidate for the molecular sensitive detection, molecular imaging, and the novel carrier for the targeted drug and gene delivery. You are acquainted with the targeted drug and gene delivery system in the previous lecture. In the as well as it is also applied for the targeted photothermal therapy. The nanoparticle in the form of nanovesicle, which is surrounded by a membrane or a layer, hold convenient surface that is spherical, cylindrical, plate-like for the molecular assembly of the inorganic or polymeric material and the drug. So the size distribution of the nanoparticle become extremely critical and play very significant role for the penetration through a pore structure of a cellular membrane. The biomarker for occupying their distinguished color required efficient fluorescence probe which depend on the narrow distribution of the average particle size. So these narrow size particles emit a very wide range of the wavelengths. The multifunctional nanoparticle core with several luminescent layers like magnetic nanoparticle can be detected and manipulate the particle. Thus, the nanoparticle provide combined platform of the biotechnology, nanotechnology, and information technology, facilitating molecular, biochemical, and biological process. For example, genetics and pharmacogenomics application. In the surface morphology of the nanoparticle can be redesigned to reach at their target like blood-brain barrier and dermal tight junctions more efficiently, overcoming the efficacy issue of the drug on the physiological barrier. In case of malignant tumor, nanoparticle with leaky vasculature structure may penetrate the lesion. So we have also discussed about the nanoparticle can be used for the development of wound healing ointment as well as wound healing. Uh, it has a wound healing property and it can be used for the uh, various pharmacodynamic aspects. So nanoparticle may contain natural and synthetic polymer for coating an inorganic core of paramagnetic material. So there are some nanoparticle and we will evaluate their application in the drug discovery process. So the first example is of gold nanoparticle. The gold nanoparticle is a nanoparticle of the gold which are the most typical used nanomaterial in the diagnostic. So they have many different use as property. So they may be extensively utilized as a connecting factor and to construct biosensor to hit upon element of the DNA. Rather than a fluorescent molecule, a gold nanoparticle can be attached to an antibody and other molecule consisting of the DNA. It can be conjugate with the protein and it can be conjugate with the antibody. So also, it can brought to the nanoparticle to supply the barcodes. So the gold nanoparticles are very chemically reactive and make excellent catalyst at the nanometer scale. So recently, gold nanostructure have found a very important role in a wide variety of applications, including bioimaging, drug delivery, toxic gas detection, biosensor, and to demonstrate the multi-photon absorption precipitated luminescence. Also, a quantum dot is also a nanoparticle which is used for the drug discovery process. The quantum dots are regarded as a unique class of fluorescent labels with the unique optical property such as high brightness and long-term colloidal and optical stability. These are suitable for optical imaging, drug delivery, and optical tracking. The fluorescence immunoassay and other medicinal applications. The unique optical property of quantum dots allow one to investigate the real-time dynamic event in the living cells. And such events include interactions between intracellular proteins, the mechanism of the intracellular signal transmission, and cell growth. Some benefits and downside have been investigated. More suitable optical residence compared with the natural dye, like incredible imaging outcome using organic dye. Absorption of numerous drug molecules for an extended time frame. The conjugate of the nanoparticle and DNA protein is also have a wide application in the drug discovery process. So the conjugate nanoparticle DNA protein hold great promise in biomedical applications. Many diverse strategies have been developed to conjugate nanoparticle and DNA proteins to assemble and purify nanoparticle protein link. First, stable and biocompatible nanoparticle are synthesized. Then, conjugate of the nanoparticle to the DNA protein is then achieved via two different approaches that do not require heavy chemical modification or cloning. The cysteine gold covalent bonding or electrostatic attachment of the nanoparticle to charged group of the protein. So co-functionalizing of the nanoparticle with PEG thiol is recommended to help protein folding. Finally, a structure characterization is performed with the circular diachroism, as this spectroscopy techniques has proven to be effective at examining the protein secondary structure in a nanoparticle protein conjugates. In general, functionalization of nanoparticle with proteins such as antibodies can be achieved by direct chemical covalent conjugation or electrostatic interactions. The semi-artificial conjugate of the nucleic acid and protein by both covalent coupling chemistry or else through the non-covalent biomolecular recognition system, which include the receptor ligand of complementary nucleic acid for immunological detection assay. Next is the nanochips. So nanochips is also developed using with the help of nanoparticles, and the nanochip system is developed by utilizing the electronically enhanced hybridization of complementary DNA strand, integrate advanced microelectronics and molecular biology into a platform technology with broad commercial application in the field of genomic diagnostics, and has achieved 100% accuracy in the detection of the nanoparticle. This technique helps in investigating DNA sequences or the pairing of the separated strand of the DNA with complementary DNA strand of the acknowledged collection that act as a probe. Currently, DNA chips is known as DNA microarray assay that employs the energy of a digital contemporary that separate DNA probes to unique wave sites at the array based on charge and the size. So then test sample, for example blood, can be analyzed for the identifying DNA sequences via hybridization with these probes. Microfluidic. Microfluidic is an emerging tools and a newly developed tools which is also called a lab on chip. It is a modern science of fluids on the nanometer scale. So the nano diagnostics involve microfluidic or lab on a chip structure in which the combination of numerous approaches of the DNA analysis is mixed on an unmarried chip composed of unmarried glass and silicon substrate. Accidentally, it is a subdiscipline of the fluid mechanics as the fundamental equation describing the physics of the fluid at larger length scale are identical to the equation underlying the microfluidic. In the general, the microfluidic biosensor platform offers numerous advantages compared with the other traditional methods such as ultracentrifugation, electrophoresis, nuclear magnetic resonance, chromatographic approaches, etc. for the detection of biospecies molecules. This device have comparable of measuring aqueous reagent and DNA containing answer, mixing the solution together and amplifying or digesting the DNA to shape discrete merchandise. This tiny possibility for the utility of the nanotechnology in the healthcare and for the development of personalized remedy seems like amazing. So microfluidic, this is all about the microfluidics. So today's summary of the lecture is the nanoparticle is a very small particle. It has a diameter of 1 nanometer to the 100 nanometer, and this is the we also learn about the nanobiotechnology. Actually, nanobiotechnology is a science, is a discipline of the combination of nanotechnology with the biological sciences. We also analyze the significance of nanobiotechnology in drug design and drug development process. We have analyzed the gold nanoparticle in the drug design and development process, the application of quantum dot for the drug discovery, the conjugate of the nanoparticle and DNA protein. We have also investigate about the nanochips, the application of nanochips in the molecule as a molecular diagnostic and the disease diagnosis process. So microfluidic device is also a fluidic discipline and emerging science that provide us for the rapid detection of any disorder or any biomarker. You can also made a lab on chip. You can also made a organ on chip for the rapid analysis and screening of the compound toxicity as well as the drug toxicity of the using a suitable assay. So this is the conclusion of our today's lecture. So I am giving you some question for your practice. So I am supposed that you should work on this question in your home and you just explore these questions and using by searching the internet, Google, and you can also read several research paper, review paper for the answering of this question. So first question is that tabulate the different type of nanoparticle. Second question is the examine the limitation of the nanotechnology in the drug discovery process, which I have not discussed, but partially discussed in this lecture. So you just outline the ethical issue related with the nanobiotechnology. So by using this